There we go. Hi, everybody. Good morning. Thank you so much for joining us, both here in person. I think you get extra sort of kudos for coming out in the rain, which is a very rare event in Austin. So thank you for being here. And to everyone online, thank you so much for joining us. We're really excited today to host a panel discussion, Biological Aging, Conceptualization, Construction, and Considerations. I'm Lauren Gaydosh, and I'm thrilled to serve here in the role of organizer and moderator for this amazing panel. So before we get started, I just want to thank the Center on Aging and Population Sciences for the support of this event. So thank you to our director, Deb Umberson, and our director of research, Karen Fingerman, who are both here, for encouraging me to organize the event and for providing support. I also want to give a thanks to Rita Stramel, our new program coordinator, and to Stacy Brody, our previous program coordinator, both of whom have been really essential in planning this event and for hosting our guests. So thank you all. Please join me in thanking them. So in my role, what I want to do first is just provide us with a very brief introduction to the concept of biological age, which will hopefully serve as a background and then allow our talks to delve more deeply into the challenges, complications, and considerations in this area of research. I'll then introduce our panelists, and they'll each talk for about 30 minutes, and then we'll have time for lots of discussion and questions. So for those who are here in person following the panel, you can join us next door for lunch and informal discussion as well. So please be welcome to that. Our discussion today centers around biological aging, which is a concept taken from geroscience. And so in brief, we know that individuals age, they face increased risk for a myriad of chronic diseases and morbidities. In fact, if I told you that you could only have one piece of information about a person in order to predict their likelihood of death or cancer or heart disease or dementia in the next year, you might choose to know their age, right? And chronological age is an incredibly powerful predictor and risk factor for health. Relatedly, biological aging is the gradual and progressive decline in the integrity of bodily systems that occurs with advancing chronological age. So critically, while related to chronological age, biological aging is distinct and represents underlying biological variation. So for example, at the end of this seminar, we'll all be 2 hours older, right? In chronological time, but not everyone will experience the biological consequences of this passage of time in the same way. Our rates of biological aging likely differ depending on a variety of factors. This geroscience perspective on biological aging really represents a sort of shift, right? From a preoccupation with death due to single diseases to an emphasis on health span and the forestalling of all aging-related morbidities. And it's thought that aging biology is driving a lot of these different diseases, but is actually malleable. And so if we can reduce the rate of aging, we'll prevent the onset of disease. Biological aging is thought to involve the accumulation of molecular changes occurring at the cellular level, which in turn lead to declining system integrity across a variety of biological systems. For those of us who are interested in health disparities, this concept of biological aging might sound familiar, right, to our conceptualization of the physiological wear and tear of repeated activation of the stress response system and related concepts like weathering and allostatic load. So how is the conceptualization of biological aging similar to or distinct from what we know about stress and its physiological consequences? Moreover, as this process of biological aging is a life course accumulation of gradual change, how do early life and childhood disparities shape aging as a life course process? So to help us consider these types of questions, we are very fortunate to be joined today by Professor Greg Miller. Dr. Miller is the Lewis W. Menck Professor of Psychology and co-director of the Foundations of Health Research Center at Northwestern University. He will discuss his foundational work on stress and health and on the life course and health disparity implications of exposure to stressors in early life. As biological aging has gained traction as a useful concept in the study of aging, technological advances in the collection and measurement of biological specimens and cellular and molecular processes have also allowed for new developments in the measurement of biological age. So there are many approaches for measuring biological age using biomarkers, which essentially aggregate age-related biomarkers using some sort of algorithm or statistical method. And there are several approaches to constructing such biological clocks or measures of biological age, particularly using DNA methylation. And our methodology session this afternoon, led by Dr. Kira Thrush, will provide a hands-on tutorial of some of these approaches. As biomarkers of aging are increasingly collected in large nationally representative studies and measures of biological aging are constructed, what challenges arise? How do these measures of biological age perform in diverse nationally representative samples across the life course, both in the US and across the globe? What have we learned about the measurement of biological age and its added value in understanding population aging and health? So I'm thrilled today that we are joined by Professor Jessica Faul. Dr. Faul is a research associate professor in the Survey Research Center at the Institute for Social Research at the University of Michigan. Dr. Faul will discuss her work on the measurement of biological age, drawing on her expertise as co-investigator of the Health and Retirement Study and co-director of the Biospecimen Laboratory at the Institute for Social Research. And finally, one potential promise of advances in the conceptualization and measurement of biological age is that more precise measurement earlier in the life course can help identify and address health disparities before they emerge. So measures of biological age have the potential to allow us to more fully develop and test biopsychosocial models of health outcomes. In the last decade or so, we've witnessed a proliferation of biological clocks, but they are oftentimes constructed using clinical or volunteer samples that are biased, particularly regarding their socioeconomic and racial composition. So what are the implications of work on biological aging for health disparities? How can we advance the field of biological aging in ways that promote health equity? So I'm very pleased today that we are joined by Professor Lauren Lee Brown. Dr. Brown is an assistant professor in the Division of Health Management and Policy at San Diego State University. And Dr. Brown will discuss her work on biomarkers of aging in diverse populations, as well as her innovative work in social genomics that has generally taken on themes of measurement, methods, and sampling that more accurately depict the aging experiences of Black older adults. So without further ado, please join me in welcoming our distinguished guests and allow me to invite Dr. Miller to share his thoughts. Thank you.
All right. Do I need to flip on my mic or am I good? I'm good. All right. Let me start by just thanking Lauren for organizing such a fantastic day. I'm really excited to be here and for thanking UT Austin for all the hospitality. It's great to be back. This is actually just about 3 years after the last time I was here, right before the pandemic. And so it's wonderful to be back with you guys. So when Lauren asked me to talk, the first thing I said is like, I'm not an aging researcher. I study kids. I study childhood health disparities. I don't know anything about aging. And she said, still, I think you can add something to the panel. So I'm going to talk about our work on stress, and particularly the immunology of stress. And what I've tried to do is build the talk around some tough lessons that we've learned, some wins and some losses. I'm happy to be interrupted with feedback, with questions, whatever you want. So go ahead and raise your hand or don't and interrupt me if you like. So let me just give you a quick overview. I'm going to start really quickly talking about predicting and understanding. I think all three of us will have some version of a slide like this, so I'll try to be quick. Then I'm going to talk a little bit about framing biosocial research questions, a couple of different approaches to doing this. I'm going to talk about some of the perils in trying to model basic science in human studies, and then some examples of how things aren't always quite what they seem. What I call the multiple layers problem. So this is the slide that we'll all have in some version, right? The big challenge for us as biosocial researchers is to balance prediction versus understanding, right? And you can think about a lot of the common biomarkers that are used in stress research, and I presume in aging research is falling somewhere in this two-dimensional space. So you've got something like epigenetic aging, which has been optimized for its actuarial value, but the mechanistic understanding it provides of what's actually happening in terms of disease pathophysiology is pretty limited at this point, right? There's a lot of ongoing research. You can contrast that with something like C-reactive protein, which we have a little better understanding of what it does from human clinical studies and from animal studies, but it's not as actuarially strong. It's good for heart disease and diabetes, but nowhere near as good as epigenetic aging and overall mortality. You can take something like atherosclerotic plaque, right, which you can measure in people's coronary arteries. Gives you great mechanistic insight about what's going on, and it's pretty good actually at predicting whether people are going to have a stroke or an MI in the short term. And you can take things like interleukin-6, which is a little more mechanistically involved in heart disease than CRP is, but actually doesn't predict clinical outcomes quite as well as it should. You can take something like telomere length, salivary cortisol, right? Probably in the lower quadrant, both in terms of actuarial value and mechanistic insights. So we're all trying to sort of balance getting into this upper right-hand corner or as close to it as we can when we're doing our research. That's the ideal, right? You have biomarkers that tell you something you want to know clinically and tell you something you want to know about process. This turns out to actually be a three-dimensional problem for most of us, right? Because the other relevant axis is how easy it is to do this, both in terms of acquiring the specimen you need and finding a laboratory that can do the measurements, right? So we often find ourselves juggling these two things, and stuff moves around quite a bit in this three-dimensional space in terms of its desirability for biosocial research. So I think cortisol stays about where it was, which is that it's really easy to do, which is why people do salivary cortisol. But it probably doesn't tell us much in terms of clinical outcomes, and it doesn't give us a whole lot of mechanistic insight, as I'll talk about later. You take something like plaque, right, which is really desirable in two-dimensional space, but it's really invasive, right? You have to put people through CT scans repeatedly to do it well. That's not possible or pragmatic for a lot of population-based studies. And then you have things like epigenetic aging, which is getting easier and easier in terms of collection and assay and procedures. CRP still, I think, in many ways kind of wins the day when you put all three of these dimensions together. And I think keeping that sort of tension in mind as you design your studies, as your graduate students and postdocs and faculty members trying to think about what do I want to study as a biomarker, it's not an easy balance. I don't think there's any marker that is perfect in all three of those spaces. You're always giving and taking some, and it's important to keep that in mind. The next issue I wanted to cover was framing biosocial questions. So I think we all agree that things work in generally this fashion, right? That you have different levels of analysis embedded within each other. If you take Bronfenbrenner's approach, you can think about social structure, policies, and practices at the highest level of an organization and embedded within there neighborhoods and communities, and then families and peers, and then individuals. And then you get to the biological side, and at the most fundamental level, at least in the kind of work we all do, you've got things happening at the level of the genome and the epigenome, and that affects what happens to cells and physiological systems and organs and tissues and disease. So if we all take this general sort of organization of the social and the biological for granted, the question becomes, how do you best study this? How do you best model it? And I think there are two different approaches to take here that are important to consider when you're designing research. And for those of you who have been to our week-long biological approaches in the social sciences workshop at Northwestern, you'll recognize this slide, and you'll recognize that we actually have a debate to open our conference where Tom McDade talks about the advantages of the population approach, and Edith Chen, full disclosure, my wife, talks about the bottom-up clinical approach and the relative merits of these two things. And I wanted to go through some of the points they make here to illustrate for you I think what's valuable about it. So the clinical approach, which is the one that Edith and I have taken our whole careers, I think in part because we've been more enmeshed in the biomedical world as clinical science, as clinical psychologists, is that you start with disease, right? You start with a disease that patterns racially or socioeconomically. And then what you try to do is take the basic science around that disease that's developed from animal models and from human translational studies. And you develop a series of mechanistic hypotheses that are specific to that disease. And in your research, you try to layer contextual variables onto each of those steps, right? And what you get out of that, if it works, is you get a medically plausible model of how higher level structural factors, community factors, family factors, and individual factors affect various stages of the pathogenesis and course of illness, right? And from a biomedical perspective, that has some advantages. The population-based approach, which I'm guessing most people in this room are more familiar with, doesn't have that necessarily goal in mind, right? So here you start with the broader social context, and then you do what Tom McDade has done so fabulously his whole career, which is you borrow clinical biomarkers that have been developed in epidemiology and clinical medicine, and you implement them in representative population-based studies, and you link context with those biomarkers. And the goal here, at least the way Tom describes it, I can imagine other people having other goals, is to really expand theories of human variation, right? To take human biological variation out of the restricted realm of medicine and to put it into the social sciences in a broader framework. And also, as Lauren pointed out, understand non-disease specific outcomes, right? To understand mortality and to understand human well-being in a more general sense. Both of these approaches have real pros and cons, in my opinion. So the pros of the clinical side is I think it's easier to get to mechanistic insights, right? To mechanistic insights in the way that biologists would think about them. And you have the potential in doing this to develop findings and models that are compelling to basic scientists, compelling to physicians, compelling to reimbursers, right? And to potentially influence the way healthcare is delivered, the way disease is thought about and managed. And given who the stakeholders are in the United States right now, I think that's not a trivial benefit if it works, right? We still get almost all of our healthcare from physicians in clinics. They hold the reins of power. There's some value in, I think, working in their system. The population-based approach is your results are much more generalizable, right? You're not talking about a specific disease, and you have the potential to much better illuminate fundamental causes that generalize across context, generalize across health problems, generalize across the life course. The downsides are almost the opposite, right? The clinical approach, it can lack generalizability. You might develop a really great biologically plausible model of how social context affects asthma, which is what I'll show you next that we do, but it probably doesn't generalize beyond asthma, right? And it can involve much more intensive and invasive biospecimen processing that's not always feasible in a larger population-based framework. The downsides, I think, of the population approach is that you often have somewhat limited control about what goes into big studies, right? There's only so much that's feasible logistically and financially, so you might not be able to get measures that are quite relevant to your hypotheses in. And I think the mechanistic insight, at least from the biological perspective, can be harder to come by in this approach. Because I'm guessing most of this room has taken or is more familiar with the population-based approach, I want to talk a little bit about the clinical approach to just give you a feel for the way we've tackled this, and hopefully in doing so convey some of the process, if not the advantages. So we've been very interested for the past couple of decades in childhood asthma, which, as you may know, patterns very strongly by socioeconomic status. These are some data looking at kids with asthma, looking at missed school days, hospitalization, ED visits as a function of their household income being at the percent of the federal poverty line. As you can see, there's a clear linear gradient in all of these outcomes. And these are interesting data. These are from the US, but we lived for 10 years in Canada before we came to Northwestern. And the data look identical in Canada, despite the fact that in British Columbia, there is universal access to healthcare that costs $96 a month for a family of 4 to have as much healthcare as you want. So it really isn't, for the most part, an access to healthcare issue. These are social and psychological factors that are disproportionately burdening low-income kids with asthma and creating disparities in the course of their disease. And we really worked hard over the years to try to understand that. So how did we do that? We started with a basic science model about what complicates asthma. For those of you who have it, you'll recognize these slides, or at least the experience of them. So this is a normal airway. As you can see, there's lots of room in the middle for gas exchange to happen, right? This is an airway of somebody with asthma that's not having an acute attack in the middle here. And what you can see is that the lumen, the place where gas exchange occurs, is much more narrow than in the normal airway. And you can see that there's a lot of red stuff around the lumen. And that red stuff is basically inflammation, right? It's cells that have moved in there, it's fluid that have moved in there, and compress the space that air can flow through. And then when an asthma attack happens, you get even further inflammation, and you also get the smooth muscles around the airway tightening up, and so you get constriction, right? So asthma is fundamentally about airway inflammation and airway constriction, and it's reversible. And what causes that? At least in the allergic forms of asthma, which are the most prevalent ones, that happens because you get exposed to allergens. The most common ones are cockroach poop, cat dander, other sorts of pet allergens. And immune cells in the airways called dendritic cells take up a little piece of those allergens, and they present that allergen to T cells. And those T cells start both what's called an early phase and a late phase response. And the early phase response involves telling B cells to make antibodies, and those antibodies bind to mast cells, and those mast cells and basically those antibodies bind to the mast cells and cause a bunch of chemicals called histamines to be released. And that is why asthma patients often take antihistamines and other drugs that block this early phase response, which results in a bunch of mucus accumulating and plugging up your airways. And then there's a late phase response that involves cells called eosinophils about 5 to 7 hours later, going and reinforcing those actions and making things even worse. And this is why asthma attacks can persist not just in the couple of hours following an exposure, but for a couple of days, depending on the patient, right? And all this stuff is coordinated by these things called cytokines. So you may have heard of interleukin 4, 5, 13, right? These are all signaling molecules that immune cells release to communicate with each other. You can think of these Th or T helper cells as the quarterbacks of the response or the conductors of the orchestra. They're the ones that send out signals that tell what other cells what to do, and those signals are these interleukins. And so what we've done historically is try to develop a model that goes hierarchically like that top-down, bottom-up model, but that's specific to asthma. And in a general sense, we've been interested in how policies and practices shape economic hardship at the family level and how that affects life stress that kids with asthma are or aren't exposed to, and how that stress influences gene expression in the immune cells that respond to allergens. And in turn, how that altered gene expression changes the way that cells behave, and how that changes the airways, and how that results in changes in asthma signs and symptoms. So this is sort of how we're going from top to bottom. This is a figure that tells you a little bit more about the details of what we're doing. But the basic hypothesis is that what life stress does is change the way that immune cells respond to allergic triggers. And to test that, we spent a lot of years trying to develop an in vitro model in a test tube where we take kids' immune cells, we take lymphocytes in green there, and in a test tube, we expose them to a substance that activates them and makes them think they're in the presence of an allergen. And we put that in an incubator for 6 hours in a laboratory, and we look at those immune cells' production of these cytokines, these signaling molecules that we know promote an allergic immune response that eventually clogs up the airways, right? So this is our outcome here in a test tube. And we're also interested in the hypothesis that part of what stress does to kids with asthma is make that cytokine response go into overdrive. And the other part of it is that it gets really hard for that response to shut down, right? We have this hypothesis that stress makes those immune cells kind of tone deaf to things that turn down the cytokine response. And what turns down that cytokine response? It's actually cortisol, right? So it's cortisol that's released inside the body, is an internal regulator of that immune response. But if you're a patient with asthma or any autoimmune disease, you also know that your physician often prescribes clinically synthetic versions of cortisol, right? Hydrocortisone, prednisone, beclomethasone, there are multiple variants, right? And those are just much higher dose versions of what your body will make. And so we hypothesize is that in kids with asthma, stress will actually make these cells not pay attention to the inhibitory effects of that molecule. And we can model that in a test tube by adding cortisol essentially to the cells and the stimulating molecule, and then we can measure production of those cytokines. And when we do this and we look at a variable like family income, what we see is fairly linear relationships that in kids with asthma, to the extent that their family has higher household income, or this is liquid household savings, you see less production of these inflammatory cytokines that cause airway inflammation when the cells are stimulated. And you see it both with these sort of late phase and earlier phase cytokine responses, right? And this is independent of the severity of children's asthma, independent of the medications they're taking. You know, it's controlled for these things as best as possible. And you also see that when we add cortisol to the mix, trying to block that cytokine response or model what happens with medication, that kids with higher income their cells tend to listen to cortisol more. Cortisol does a more effective job, implying that our hypothesis about the efficacy of anti-inflammatory treatments is lowered in lower income kids. So this is an example of kind of the first stage of our research. We spent about 10 years to make those 2 slides. And lots of the Canadian government's money. And it was the first step, right? So we're linking here economic hardship with changes in cellular responses, right? And if you buy this clinical approach and you want to take the next step, what you would do then, or what we did is think, okay, you can either go up or down, right? You can dive into some of the molecular pathways that might explain why you've got changes in cellular behavior, or you can go the other way and try to understand what implication those changes in cellular behavior have for the airways, right? Either approach is reasonable. We did both, but I'll tell you a little bit more about this one. So the next thing we did is, in the following study, we collected immune cells from kids with asthma, and we separated those cells out. As I'll talk about in a minute, if you collect a blood sample, you've got a mishmash of very different types of cells. And so what we did is physically separate out the ones that are important for asthma, and we did gene expression studies to look at messenger RNA and look at transcriptional pathways that are important to the activation of cells. So these are gene expression studies specifically in these T helper cells that are sort of the quarterbacks of the immune response and the ones that really drive the early and late phase asthma response. And we did it in a sample of kids where we tail sampled for high and low family income. And the details here, I don't want to go into because I'm already going over time. But the point is that the next step we took was to identify some of these transcriptional pathways that differed by SES in the key cellular players in this response, and then associate them with measures of cellular behavior, right? And that was the next step in the project. And our approach would be to go here again, and then connect it to asthma signs and symptoms, and also to move up to understand these boxes and unpack them more over time. And that becomes the research program, to sort of go up and down in the hierarchy. This is a very different approach, I think, than the population-based approach. But as I hope I've conveyed, it's got some advantages in terms of really focusing in on specific biological pathways that are relevant to diagnosis and treatment of asthma. But this isn't always such a great approach in some ways. I want to talk about the caveat is that although there's often great basic science to guide this work, it can be really hard to model well in humans. So you think that what you're doing is following kind of the hardcore, elegant biomedical science that gets into Nature and gets into high-profile journals, and then you try to do it in humans, and you realize, oh, this only works well in mice models or in non-human primate models. So telomeres, I think, are a great example of how hard this is to do well, right? So you guys probably all know telomeres are these caps at the end of chromosomes. I know that Lauren's going to talk some about this, so I don't want to go into a huge amount of detail. But telomeres are key in the pathogenesis of some cancers and some aging-related diseases. They have a number of functions. They've been most interesting, I think, in the biosocial world because of their role in cellular aging. And just to go really quickly, Lauren will probably talk about this more, right? So there are these caps at the end of chromosomes. Each time a cell divides, the telomeres shorten, right? And that eventually these telomeres shorten enough that the cell stops dividing. And this is a great check on cells becoming immortal, because when cells become immortal, they keep dividing, and that results in cancers and tumors. That is the briefest overview of telomeres I will ever give. And these have been tremendously interesting to stress researchers. You can see this was a paper in Nature a couple of years ago, or I guess a decade ago now, that Alyssa Eppel and Liz Blackburn wrote. For a study of human stress to make it into Nature is almost unheard of. So lots of press. They had this book that was a New York Times bestseller on the telomere effect. If you look at, just do a quick PubMed search at the trajectory of publications on stress and telomeres, that's a pretty steep line. Alyssa's first paper was like right about here in PNAS. There's been a lot of interest in this stuff in our field. Here's where it gets tough in human studies, right? Is that we, in human studies, collect blood, sometimes from the antecubital vein, many times from capillaries in the fingers. And when you do that, as I said, you get a real mismatch of cells. So this is what the typical white blood cell composition of human venous blood looks like. About 65% of those cells are granulocytes, about 5% monocytes, about a third are lymphocytes. Why does that matter? That matters because these granulocytes and monocytes, which make up 70% of your blood, they don't divide at all, right? They're born, a few days later, they die. They're completely gone. They don't replicate. Lymphocytes do. Lymphocytes divide, right? Monocytes and granulocytes also, basically telomerase, the enzyme that allows telomeres to stay at the ends of chromosomes during division, there's almost no telomerase activity in 70% of the cells you get in a typical blood draw, only in these 30%. And so the question becomes, if 70% of the cells in the blood sample you collect die within a week and they never divide, what's telomere length telling you, right? And you can do all the correction you want for differential cell composition. You can deconvolute the heck out of this. But the fact is that almost all the variance in your sample is from cells that don't divide, where replicative senescence and aging isn't really an issue. They're all going to die. They're all going to die very quickly. And it's possible that the signal you're getting in your mishmash of cells is telling you something about a progenitor cell in the developmental pathway. So if you look at how immune development happens, or the hierarchy of immune development, you have these things called hematopoietic stem cells, right? These are the grandmother cells of all your blood cells, right? And they divide into what are called common myeloid and common lymphoid progenitor cells. And those common lymphoid progenitor cells divide into T cell progenitors, which can become T cells or B cells, and the T cells can subdivide. As you can see, there's a whole hierarchy here. It's possible that when you're reading out the telomere length, or for that matter, the epigenetic signature of a bunch of different cells, say in this lineage, that it's really telling you about what's happening in that grandmother cell, right? That would be really cool if it was true, right? But we don't know that. Nobody knows that. Nobody knows whether that's true or not. It would really be fantastic if in a tube of blood, you could tell what was going on in these stem cells. It's possible, but we don't know. Compounding the problem is the fact that all these different types of immune cells that you grab in a sample of blood, they all have differences in their native telomere length, right? So some of these cells have lots of really long telomeres, and some have really short telomeres. Nobody really understands why that is. Further compounding the problem is that the composition of your pool of white blood cells, it changes with stress. So if you put somebody through a very brief Trier social stress test, like example, the number of lymphocytes you find in their blood drops by 20%. Monocytes and granulocytes rise. It's not like these cells die, but the lymphocytes go and hide in your lymph nodes in your spleen, and these cells come out of your lymph nodes in your spleen and show up in blood. So the very act of measuring stress in some ways or inducing stress changes the population of cells. These cells differ in their telomere length, and it becomes a really hard problem to solve. So the same concerns, as I said, could apply to epigenetic studies in blood, right? Because different cell types have different CPGs that tend to be methylated, hyper or hypomethylated. And again, most of these cells don't divide. So if you're thinking about epigenetics as a way to understand, say, the biological embedding of early experience, but almost all the cells you're studying are ones that turn over within a week, it gets very hard to make a case that these cells are telling you something about a biological memory of an earlier experience. Unless, again, they're telling you about something way back in the developmental history of stem cells. And we just don't know that. It's possible. It would be awesome if it was. All right. So what's a poor social scientist to do? There are ways that you can separate out cells, right? So this gets you back to the clinical approach, some of the stuff that I talked about earlier, where you can say up front, based on your understanding of the pathogenesis of some outcome you're interested in, these are the cells I want. They do divide. I'm going to literally pick them out of my blood sample and do my epigenetic studies or my transcriptional studies or my telomere studies in these cells. That's totally plausible. It just requires money and time. The second problem with this clinical approach is that stuff's not always what it seems. There are so many layers of biology, and I think our understanding of those layers is pretty naive, at least mine is. So here's a case study with cortisol. We spent decades gathering like gallons and gallons of spit from people. Like, I cannot tell you how much spit I have collected over the course of my career to try to understand how stress affects the HPA axis and the output of cortisol. And I'm not the only one. When Clemens Kirschbaum published this paper in 1994 on using salivary cortisol, psychologists just went nuts. And we're 30 years later and they're still going nuts and everybody else is getting in on the act. Here's the curve of publications. Here's how cortisol works, right? So cortisol gets released from the adrenal glands. It goes into circulation and it enters tissue. So let's say it enters your lungs or your lymph nodes or your spleen, and then it has to enter individual cells, right? To do anything, cortisol has to get past the cell membrane and inside the cell. And by changing the way that cells behave, generally in terms of the genes they express, cortisol is going to change the way that organs behave, right? But it's got to act at that cellular level first. But getting into the cell for cortisol is no easy trick and not a given. In fact, if you learn a little bit about the cell biology of cortisol, the first thing is that cortisol travels through the circulation bound up to this thing called cortisol binding globulin. Like 90% of cortisol is bound to this thing. And if it's bound to this thing, it actually can't get into the cells. The cells can just deflect it away. Now you can measure unbound cortisol. In fact, the stuff in saliva is unbound. So what you're measuring has the potential to make it inside the cell. The problem is that cells have all these defensive mechanisms to push cortisol out. So there's literally this thing called the MDR, multi-drug resistance pump, that once cortisol comes into the cell, it just sends it right back out. If it makes it past that MDR pump, there are these enzymes that also can change the structure of cortisol so that it's not able to move into the nucleus. If it makes it past all those things, it has to bind to a receptor inside the cell. That complex has to divorce all these chaperone proteins, heat shock proteins, and other proteins. And it has to go through about a dozen steps before it makes it into the nucleus. And those steps are reasonably well understood at this point. But the odds of like cortisol from here making it into the nucleus and changing cellular behavior turn out to be much lower than you would think. And they depend on the time of day and how saturated those receptors are. If it happens, then cortisol can move into the nucleus. It can change gene expression patterns by binding to glucocorticoid response elements, all the stuff that you're no doubt familiar with at this point. So how does this play out? We did studies back when I was in Vancouver of one of the most stressful experiences that I've seen up close as a psychologist, which is studying people whose spouses had been diagnosed with glioblastoma. So glioblastoma is a very serious brain tumor. At the time that we were studying it, the median time to death from diagnosis is about 10 months. And during this 10 months, patients are going through a series of very disfiguring treatments, not only brain surgery, but radiation to the brain, dramatic changes to their well-being, to their personality. And so we followed spouses of patients shortly after diagnosis for a year, trying to understand psychological well-being, things that fostered it, things that detracted from it, and the implications for immune cells. And what we found is that with all these layers of regulation, whatever caregiving stress of glioblastoma did to cortisol was really mysterious. And so let me show you some slides. So this is gallons and gallons and gallons of spit from caregivers and controls. This is something like 15 days of saliva collection 5 times a day. And as you can see, bupkis, right? Probably the most stressful experience we could think of studying. And there are just no reliable differences in cortisol at any point of the day in these samples. And then we went and used a technique called flow cytometry to measure the amount of receptor for cortisol that immune cells had, thinking like, okay, if there aren't differences in the amount of cortisol, maybe there are differences in the receptor that's available to bind it. It turns out that that's wrong too, right? So nothing in terms of receptor levels, which makes you think, what's the deal? What's going on here? But then if you start poking the cells around, you find that actually there's something different going on here. So these are experiments where, like I told you in the asthma work, we take people's immune cells, we put them in a test tube with bacteria, and we look at, when they're exposed to bacteria, how much inflammation they produce in terms of cytokines. And then we add cortisol to sort of shut that process down, right? And when you do that, you find that the caregiver cells actually are much less sensitive to cortisol's anti-inflammatory effects than the control cells are. So even though there are no differences in cortisol levels, even though there are no differences in the receptor, the cells are responding very differently to cortisol. So something's going on here in the crosstalk between cortisol and inflammation in immune cells that isn't evident just in levels of the hormone and isn't evident in levels of the receptor. And then we can pull those monocytes out and we can do gene expression studies, and we can do bioinformatic analyses that tell us how active genes are that we know cortisol can activate or deactivate. And we see differences in that readout too. And so what we conclude here is that caregivers' immune cells are kind of tone deaf to cortisol signals, even though there are no differences in cortisol to be found in their blood, or it turns out in their urine, or it turns out in their spit. And there are no differences in receptors. And yet something more profound and deep is going on in sort of the machinery of the cell. And for me, this all illustrates just there are these multiple levels and multiple layers that when you unpack them, they seldom converge. And that to really understand the biology of this stuff, one has to be dedicated to sort of going piece by piece and understanding the full picture. So in closing, what have I learned? This stuff is really hard to do well. There's that famous Voltaire quote that I won't give here. I can't remember the details, but it is really hard to do well. You know, that I need to think hard and often about, you know, what my objectives are and who my audience is, right? For us, the objectives and the audience are often the biomedical community, and trying to go and advocate in settings of allergy and asthma for better care that's more sensitive to social determinants of health, and to use what we've discovered about the biology to talk in the language that physicians talk and pharmaceutical companies talk. And that's been pretty successful. And to really design our studies and what we do with those issues at the front of mind. And then to be critical and humble about the biology and is telling me. I think this is probably the fourth thing, the one, after 20 years of doing this, I think every year I hope to get more critical and more humble, and the data have a way of doing that to us, for sure. So with that, I wanna just end here, and I can, if I'm over time, I can sit down and let Jessica go.
Does anybody have immediate questions? Okay, Karen, yeah, let me give you the mic so that people.
So great. Thank you for that talk. That's all I wanted to say.
Oh, that's so
I've just been
nice.
sitting-
Thank you.
Excellent. Okay, great. Well, we'll have more time for question and discussion at the end, and we'll turn it over to Jessica Faul. Thank you so much, Greg.
So I think Greg did a great job talking about the complexity of what we're up against when we want to think about collecting biological data. Really just in terms of, the samples that we're collecting and the multitudes of different kinds of cells and how those cells can behave differently in our bodies. Lauren asked me to talk about the dirty secrets of trying to collect these kind of data on a large scale in a population representative sample, and also some of the complexities that we think about when we try to integrate these things into our social science models. I didn't call it dirty secrets, I called it a look under the hood, but I think you'll get a sense of the types of things that we think about. Like Lauren said, I'm an epidemiologist by training. I work a lot with the health and retirement study trying to figure out what types of biological samples are possible for us to collect in studies of 20,000 people that are not clinically based or lab-based samples. So a lot of times we move from doing best practices in the lab to collecting out in the field. That changes a lot about what we can do, how good those samples are, the relative validity and reliability of what we're collecting. So that's a big challenge and what I'll talk a little bit about today. And like Lauren said, I also run the biospecimen lab at the Institute for Social Research. So we help other studies think about, what are really good biological endpoints for them to consider, given all the factors that they may be up against in the studies that they're doing as well. So in my day jobs, I think a lot about best practices, data collection in the field, and like I said, how we integrate all of these things together. So when we think about aging biomarker goals, we want things, like Greg said, that are minimally invasive. I can't get brain tissue very easily, nor would anyone give it to me. So I've got to think about what can I get from peripheral tissues, and is that going to be relevant for the thing I'm studying? Ideally, I want something that tracks with chronological time, but might be a better predictor of outcomes than chronological age alone, like Lauren mentioned early on. I want this ideally to be responsive to interventions or lifestyle factors. I mean, clearly it's something that we would like to improve. And it should facilitate our understanding of the basic biology of aging. Those are our goals. Like Greg said, those are often hard to come by. So here's our biology primer for the day. Greg got you started, so I'll get a little bit more on DNA methylation, because that's largely what I'll talk about today. So DNA methylation, these are methyl groups that can tag DNA and activate or repress genes. That's why we're interested in them. We talk a lot about CpG sites. Those are named because the DNA methylation is where the sites where methyl groups attach to DNA happens where a cytosine nucleotide is followed by a guanine. So CpG, that's where it comes from. And this is just one type of epigenetic modification. There are a lot of other types. On here you can see histone modification. We go for DNA methylation because it turns out to be among the easier ones to study in terms of epigenetic modification. There are large DNA methylation shifts early on in development. A lot is happening in utero and right after someone's born, because these DNA methylation sites establish cell lineage. We have the same DNA in all of the cells of our body, so methylation largely is what turns something into a red blood cell or a nerve cell, intestinal cell. That's what makes it different types of cells and have different functioning in our body. And so this is critical. It's a critical part of health. And I think what we often forget is this is a normal part of what our bodies should be doing. If we didn't have DNA methylation, we would be in a world of trouble. So I think oftentimes it really gets almost stigmatized like a disease state, when in actuality, it's a health state. Like, this is what we're supposed to do. When we measure it, at a single cell level, DNA methylation is binary. It's either unmethylated or methylated. When we use these bulk samples, so samples of saliva or blood or really any types of cells, DNA methylation is a number between 0 and 1. So this is the proportion of cells that are methylated at a given CpG site. But most of what we're looking at is either methylated or not methylated. Like, the vast majority is 0 or 1 at all of these sites. So we are taking advantage of the sites that kind of fall somewhere in the middle, but that's not the vast majority of them. And this is actually fairly stable across our lifetime. So we're really leveraging this murky middle here. So based on the age association DNA methylation changes that occur relatively constantly in individuals, there are a lot of groups that have taken DNA methylation to create these clocks that measure the chronological age or other phenotypic outcome of the donor. They're usually developed using machine learning, so a supervised machine learning approach such as penalized regression, and they consider hundreds of thousands of CPG sites to build the best predictor model. I took my slide out because I knew Greg had one to really optimize this idea between biological understanding and prediction. So these clocks have nothing to do with biological understanding. We are taking 850,000 CPG sites and just trying to pull out the ones that are the most associated with the outcomes we care about. So it's solely prediction. There are dozens of different clocks, more every day, and they're all trained in different ways. So, and these various clocks have different associations with outcomes because they're trained to have different associations with different outcomes. So the first generation of clocks, the things that are on the left-hand side there, those were largely designed to predict chronological age. And then as we moved on in the field, we thought to ourselves, well, it's a lot cheaper just to ask somebody their age than spend hundreds of thousands of dollars measuring DNA methylation. Wouldn't it be great if we had something that told us something a little bit different than that? And that's this concept of biological aging. It is, the idea that there's a biological phenotype that represents aging that might be, you might be older than your chronological age, you might be younger, and that's really the target that I think we as health researchers or social scientists, whomever, whatever camp you identify in, that's the thing that I think is most interesting to us. We also talk about a concept of epigenetic age acceleration, and that's the residual of the clock values regressed on age. So we can talk about that biological aging examined without the effect of age, because otherwise it's too confounded together. And most aging clocks are moderately correlated with each other, and their age acceleration measures are weakly correlated. So this figure kind of gets at what I was talking about in terms of the perfect chronological and biological age matching versus someone like red person, who's aging faster than their chronological age suggests. And so ideally, like I said, this is the difference we want to capture. There are differences in tissue and organism functional decline, differences in the risk and actual age association with disease, morbidity, and mortality, and that is what we're hoping to capture. So already this has moved to the commercial space. You can spend a couple $100s and you can turn in your saliva sample and they will return to you what your biological age is. So that the industry has been very quick to move to this space. But the questions we have to ask is, what do we actually know about this? What do we know about these clocks' ability to predict at an individual level? So this is like criterion validity. How specific are these clocks to their training data sets? So how portable are they? If I identify them in a sample of older Italians, is that going to work here in the United States? I think Lauren might talk a bit about this. And also, what is their reliability? So we did some digging into this. So here I'm going to show you the correlation between biological age and chronological age using some of the clocks that are designed to predict chronological age. So they should be really good at this. So this is the Horvath and Hannum clocks. You may have heard about this. And we'll use HRS data from our entire sample. So well, not everyone between 51 and 90. We'll leave the oldest old out for this. So here we have Hannum and Horvath in dark green and light green. And you can see when I look at the full sample, the correlation's really high. It's between 0.7 and 0.8. That's great. You see this and you think, oh yeah, let's do it. Like this is a great measure, let's go for it. But if I start looking at progressively smaller categories of age, my correlation starts to drop. So here are 20-year age groups, 10-year age groups, and then 5-year age groups. So this is how accurate are these clocks at predicting these people's chronological age. So when I get down to those 5-year age groups, the correlation is between 0.1 and 0.2, excuse me. So like not that great. So this is sad. And it puts into question whether we can accurately use these clocks to predict at an individual level. If I look at a different kind of clock, so this is Grim age, this is one of those phenotypic clocks. This one was designed to predict mortality. Age and mortality track together, so it's highly correlated, but you can see the same pattern is there. So again, if I look at my whole sample, 40, 50 years of data, highly correlated, I get down to these smaller groups and much less correlation is there. So another thing we wanted to do was to better understand how important the specific CPG sites identified in these clocks were. So this was kind of getting at, like, is it picking up really important biological signals or what? So was there a special aging signal, or is aging a phenomenon that was affecting DNA methylation more globally? So what we did here is we took the 860,000 CPG sites we have measured in HRS on just over 4,000 people, and we created what we call random age clocks. So we took 50 random samples of 50,000 random CPG sites drawn from the total that we measured, and we trained our own clocks. So we had, we split these samples with 75% of our sample on the identification side and 25% on the validation side every time. We used the same method, so an elastic net regression, and then we tested how well could we predict chronological age using random sites. And what we found was this. We were really good at it. So this is not taking all 850. This is taking random sets of 50,000. So our mean correlation was really high, like 0.87. We were better than most of those other clocks. Again, this is the whole group. So what does that tell you? It tells you that methylation is happening across the genome and not necessarily at particular sites. In fact, most of the clocks, so this is showing you how many different CPG sites got built into each model. It varies from about 1,000 to 20,000, but on average, most of the time we were pulling data from about 18,000 of those 50,000 sites. So what that tells you is that around 36% of the sites were associated with age, and which ones are selected in any given clock are somewhat arbitrary. I mean, I think part of this was known in the epigenetic world, but that methylation is happening across the genome, not necessarily at really important biological sites, but kind of more globally. So another thing we worry about are the effects of collection on the outcome. I don't get to bring people into a lab. I don't get to process their blood or their saliva in under an hour and get it frozen. I've got to ship it in the mail. I have to do all sorts of horrible things. And this can affect both the validity and the reliability. So variation and problems can introduce themselves at any of these stages. We start all the way on the left with collection. And I put in blue, like the variables that we're keeping track of when we create data products that are marking these things. So which tissue am I using? Is it saliva? Is it blood? When I do the DNA extraction from that material, what's my concentration of DNA? For DNA methylation, this plating and bisulfite conversion is where a lot of our problems get introduced. We're moving those samples to the plates, but the bisulfite conversion is happening on a plate level, and that is something that varies a lot between plates. So here I'm keeping track of the plate, the batch, the row, the column. And then I'm moving those samples to something like this. This is the Illumina EPIC chip that we use for DNA methylation measurement. And there's variation here too, because the fluids that I'm using to help create this signal are gonna flow from the top to the bottom. So I've got higher concentrations at the top than I do at the bottom. This changes the intensity of the signal. Lots can go on here too. So we did a pilot to really try to understand, how are these components working together to introduce variation in my measurement and potentially complicate everything that I want to look at downstream. So we tested various methods of saliva and blood collection and different storage conditions. So how long did I wait from the time I took the sample to the time I processed it? So extracted the DNA and got it into the freezer. We wanted to see where does this variation come in. So generally, DNA methylation is thought to be stable, but like Greg said, a lot of these differences that we can see could be the differential survival of different cell subtypes once they're taken out of the body, and the distribution of DNA methylation that is different in these different kinds of cells. Normally, cells don't like to be outside of your body. They kind of start freaking out as soon as we take them out of their native environment. And if some of those cells are differentially affected by that traumatic process, then at the end we could have a really different composition of cells in our sample. So we collected saliva and venous blood from 15 different volunteers. We collected lots of saliva, like Greg does, and we divided these into different aliquots, and then we stored them for different amounts of time. We processed them immediately. We stored some for 1, 2, 6, or 12 months at room temperature before we processed them. And then we also collected venous blood in different types of tubes. So EDTA tubes, heparin tubes, a PAX-gene DNA tube. And we did the same thing. We processed them immediately. We left some refrigerated this time for 1, 2, 3, 7, or 30 days before processing and freezing. So this was really trying to simulate the awful conditions that we have to deal with field collection, and we wanted to see how far could we push it. We used the Illumina EPIC chip for DNA methylation. We plated a lot of duplicates, so we put the same sample on different plates so that we could understand those batch and plate effects. And so here we're looking at the effect of tissue, the effect of DNA concentration, tube type, cell composition, and storage time and batch effects, all on the downstream measurements that we're going to get. So first, to compare across different experimental factors, in order to think about this easily, we calculated a lot of different clocks, because I think this gives us a more intuitive understanding of what's happening, but also is the endpoint that we're actually going to use, so knowing how all of these things affected our endpoints was helpful. We ran a lot of fixed effect models, because essentially these are panel data, right? We've got intra-individual collections, but across multiple periods of time. It's just kind of weird in this case, but it's the same. So this is what we did. So we ran these fixed effect models to model changes or differences within individuals, and we looked at interclass correlations for each clock. So how correlated is every measure within an individual given these differences in collection? So the interclass correlation for the combined tissue is generally lower. So this is putting blood and saliva together, and it's the blue bar. Because DNA methylation in saliva and DNA methylation in blood, because we have different cell types in those samples, is not the same. But the interclass correlation is much higher for measures within tissue type. So here we're looking at how long did we wait before we processed it, but comparing just within saliva, just within blood. And so this was promising, because this was starting to tell us that all of those different factors that were different within saliva and blood didn't make as much difference as we may have been worried about. Another way to look at this is what contributes to DNA methylation variance within looking at one clock, but comparing the different types of collections that we had. So on one side, we have grim age, which is the one I told you about that predicts mortality. And on the other side is a POAM45. So this is looking at a composition of different biological measures at age 45. And so you can see here what's contributing to variance in those clocks is different for different types of clocks. With grim age, a lot of the variation was coming from batch. So I told you that batch really introduces a lot of problems for us when we have multiple plates that we're running. And here, about 60% of the variance is coming from batch, no matter if I'm looking at all within blood or all within saliva. Whereas the POAM45 clock, a lot of that variation was coming from cell composition. So the answer really here is it depends, but I will tell you from most of the clocks that we looked at, cell composition and batch were the most important factors and the biggest pieces that were contributing to our variance. So let's look at this another way, and let's see how these factors change the predicted epigenetic age. So here we have on the plot, the figure, all of our different individuals and their ages. So we had people from just over 20 to age 60. And again, we have all these different experimental methods that we're thinking about. So this first figure shows you the effects of those different types of blood collection tubes. And you can see, with the exception of heparin, which is this kind of gold triangle, most of these are clustering right around the person's real chronological age, which is exactly what we wanted. So this is telling us that heparin might be trash, but the rest of the blood tubes that we used really worked well, and they clustered really nicely where we wanted them to be. And then we looked at batch. So red here is one batch, one plate. Gray is the other. These are the exact same samples. So, not necessarily different time points, but we did, time 0 in 2 batches, time 1 in 2 batches. These gray squares and the red triangle should sit right on top of each other if we didn't have any batch effects. But this shows you that the batch effects were actually really big. All of our red triangles are clustering together. All of our gray squares are clustering together. They should all be together. And also think about the scale on this. This is a 60-year-old person that this batch is telling us is really 25. So that's a big problem. So here, the good news is that collection issues, concentration, tube type, time, these things aren't contributing to the variation that we worry about as much as batches. And the within person range, so within a batch or across batches, on average across all the different clocks, this is about a 2 to 5 year difference. So this is pretty big, right? We don't want that. We really would like this number to be 0. It won't ever be 0, but 5 years is definitely too much. Here we've plotted the same sample types, but across many different clocks. So I wanted you to see, don't worry about what the clocks are, but just see the range. These clocks, yes, they're designed to do different things, but we have, 40-year, 50-year age range on some of these. That's also of concern. The differences here are large. So the between clock range on average is between 5 to 10 years. I just want you to see this because you have to think about these type of things when you're asking your scientific questions, but what type of effect size would I expect from the thing I'm looking at, and how does that compare to the measurement error that I have going on with the sample that I can actually collect? So, and we also try to think about what can we do about this. We can adjust for cell composition, we can adjust for batch, and that helps a lot, but there's still some pretty significant measurement error that exists even after I do those things. So smart people, some of whom are in the room, attempted to deal with this mathematically. And this was a really great stab at this. So they decided to train the clocks differently to see if they could get around some of this. So they created what we now call as PC clocks. And this was a proposed method to reduce the underlying technical noise in the DNA measurement, from all the things that I just showed you. So they used the original data sets that the clocks were trained in to create principal components. And then they trained the clocks on those principal components instead of on the CPG sites themselves. So then you end up with a set of beta weights that you apply to your data with each CPG site to each PC, and then each PC to the outcome. And the idea here is that this technical variation is sort of randomly distributed across the genome. So we're trying to ignore that and try and pick up the signal that we care about a little more cleanly, ignoring that technical noise. So we set out to figure out, well, does this help us? It does in a way. It really sort of reduces a lot of the variation that we see when we look at something like, the epigenetic age that predicts that it produces, but we wanted to see, does that help us in understanding the relationship between epigenetic age and the outcomes? So we used data from the health and retirement study, again, to look at the relationship between DNA methylation and health-related outcomes, both cross-sectionally and longitudinally. We look at whether the PC clocks improved our prediction of aging-related outcomes. And then, maybe most importantly, how does that compare with all the other pieces of information we know about someone and the ability of those things to predict outcomes? So this is all work that's under review. And we looked at a lot of different outcomes, but here I'm only showing you the effect on multimorbidity and 4-year mortality. And what you can see here is that there really aren't that many differences. The PC epigenetic clocks did not significantly change the relationship of DNA methylation-based age acceleration measures on the health outcomes or mortality compared to the original clocks. So they did seem to clean up some of the noise, but they didn't help us improve our ability to find that signal that we wanted. And the authors actually themselves acknowledge that they found very similar things in the data sets they looked at as well. And then this piece that I mentioned we also looked at too, which was how does epigenetic age relate to the other things that we know about people and the ability to predict outcomes. So here, this is just an R-squared decomposition from regression predicting ADLs and IADLs, multimorbidity, cognitive dysfunction, and mortality. And we have all the things that we like to look at, age and sex and education, race, ethnicity, health behaviors, prior depression, childhood situations. And the gray bar is the grim age acceleration. So you can see, depending on the outcome, it has a different relationship in its ability to predict that outcome. And depending on what your prior was, this is either really exciting to you or really sad. So it really just depends on what you expected this measure was going to be able to do. I like to say that DNA methylation does not usurp the importance of the social variables that we know about. It's not eating up all that signal. All the relationship we know about between those social factors and outcomes is not operating through DNA methylation alone. But it also appears to be a fairly robust biomarker. So it's true that it's not everything, but it is giving us new information. So like I said, depending on how you look at it, it's really great or not. But I think there's promise there. There's promise for us to learn more, especially as we get around these measurement issues and build this into our models of aging. So in summary, because I know I went through that really fast, but understand that methylation is something that is happening across the genome. Fortunately, for those of us who have to collect data, the collection variables, so the tube type we use, the DNA concentration, and the storage do not substantially impact DNA methylation measurement, but we still have a measurement problem from the assay itself. Most of these clocks have at least a 3-year measurement error on them related to batch or cell composition, and this is big. So this means that if you want to look at DNA methylation, your meaningful effect size is going to have to be larger than that. And studying short-term change with or without an exposure or intervention is going to be super tough. Don't expect this to change in 6 months without something really severe and dramatic happening in that time period. So really don't ask too much of this biomarker too soon. And really, I would say right now, the measurement error for these types of clocks, thinking about kind of global aging-related outcomes, is currently too high for clinical applications. When you collect data, if you collect data, plate your own duplicates, put that same sample on multiple plates so that you understand reliability in your own sample. I will tell you, unfortunately, that the probes that turn out to be unreliable are different every single time we do it. It's not like there's just a set of bad probes that we can adjust out of our data. It's different every time, and that's really makes it hard and unfortunate for us. And really just temper your expectations. DNA methylation is not a silver bullet. And we always go through this, unfortunate experience every time we have a new biomarker where we expect the world of it, then we're really sad and crestfallen when that doesn't work. And then, we usually end up somewhere with a happy medium. So really, this is just, thinking about all of these things that are going on in the background when you think about your research and incorporating methylation into it. Shameless self-promotion. We have a course if you want to learn more. So we do 2 different courses, Genomics for Social Scientists. One is an introduction course, and one is actually specifically on epigenetics. So if you want to come to Michigan for a week in the summer and learn about this, learn about it from a social science perspective, do hands-on training with data, you are welcome to do so. We do both virtual and in-person formats, and there's some brochures over there if you want more, because we're actually taking applications right now for the epigenetics course. And just thank you to a lot of my collaborators at a lot of different places. So thank you.
Excellent. Thank you, Jessica. Do you have any immediate questions? Yes, please. I'm going to bring you the microphone so the people online can hear you, if you don't mind.
So in terms of batch correction, does your group typically like account for the slide and array position, or because I've always wondered how different groups what they use as a batch?
So I would say the best thing to do is definitely to adjust for batch, right? Because that's getting that the plate number. So everybody that was had bisulfite conversion together. The other really important variable turns out to be column, because these 8 wells transfer over to this chip exactly as they are in columns. So column is actually marking which chip they're on. And then the other piece of information to know is where they are on this slide. So were they at the top or the bottom? So I would say with those pieces of information, and where they are on the slide is actually row because of the way they move over. So with batch, column, and row, you know everything that you need to know in terms of, those sort of batch effects that we worry about, both from the plate and from the slide. Yeah.
Excellent. Thank you so much. Okay, thank you, Jessica. And next we'll have Dr. Lauren Brown.
Hey guys, is this on? Yeah. Good morning. It's great to be here. Lauren, thank you so much for inviting us. The coherence in this panel is wonderful, and it will keep me from over-explaining things that aren't necessary because of how wonderful the audience and as well as Jessica and Greg in terms of setting me up. So today I'm gonna add another level of complexity to what it is we're doing with a lot of the biomarker and social genomics data that we're collecting, and really thinking about the empirical ethics of what we're doing with a lot of this data and how we're applying it to diverse populations. So I'm going to use the example of salivary telomere length because that's where a lot of my research lies, but I'm going to be talking more broadly about biomarkers and social genomics and how I think telomere length is a really great blend of both a molecular biomarker that has molecular and genetic issues, as well as a biomarker that is more similar to something like a CRP and IL-6 that's derived from your biological space and not necessarily a marker of your genetic variability. So I came to this work really thinking about how Black Americans are aging and some of the unique difficulties that Black Americans face in reaching older adulthood. And a lot of my work was really grounded originally in stress and stress measurement. And as a result, this idea and concept of measurement has been something that's been really, I think, foundationally grounded in how I approach the biomarker space because of how much trouble I had in my dissertation work in thinking about stress measurement for Black populations. And there specifically, I was looking at, my dissertation was on this measure of chronic stress in the health and retirement study, where they ask about ongoing chronic stressors, but in the same question, they ask about about the appraisal of these stressors. And our Black respondents and also our Latinx respondents had very different appraisals across these chronic stressors, which manifest differently for mental health. Physical health, I don't know yet how much that's manifesting differently for physical health. But this idea of measurement for Black populations, I think, is incredibly important because a lot of what we do now is take what's done in white populations and just transpose that to every other race and ethnic group. And I think that really causes problems once I start getting into the literature and trying to apply some of this stuff to Black people and to their aging process. So that really led to me thinking about telomere length as a molecular biomarker, trying to capture the stress process. But as you guys know, there's a ton of issues in terms of stress measurement in telomere length. And I'm not quite sure if we're even capturing that stress process for these populations at all. So I like to start my talks here mostly because it just grounds the idea that aging is in a really important lens to think about health disparities. And that I think is because largely a lot of what kills Black people at a higher rate in particular is diseases of aging. So heart disease, cancer, and diabetes make it in the top 4 of the 5 things that disproportionately kill Black people over the life course. But the other reason I really like this slide is because it doesn't just ground us in the Black disadvantage narrative, which is often really the predominant and prevailing story in Black health and aging. And trying to have a more nuanced discussion about Black health can often be difficult because what we ground a lot of our research in is this disadvantage narrative and comparing Black people to white people as the gold standard or the norm of health and aging, when in actuality that may not be a super relevant comparison in the lived reality of what Black people are experiencing. And it doesn't always overlap with what they think of their own aging experience. And so to acknowledge them a lot more nuanced, I think it's important to highlight both the Black disadvantage narrative, but also acknowledge that there are a host of outcomes that we don't talk about or don't cover where Black people actually have an advantage in terms of mortality in this case. But what I think is also important is that mortality and diagnoses do not capture everything that we need to be thinking about. And this slide, I think, does that really wonderfully. This shows that the diagnoses and death rates are not exactly matched when we're thinking about disparities in health, suggesting that mortality rates for Black women are higher in terms of their breast cancer death rates. But when you look at diagnoses, you don't get that same picture. There's actual lower diagnoses. And I think a lot of that is because we often norm white healthcare norms onto Black populations. And healthcare norms are not the same across these groups. And so things like diagnoses may totally misrepresent the health and well-being of Black populations. And then you get to the mortality rates and you start to pick up, I think, what we think is more consistent with what the Black disadvantage narrative is telling us. So I'm not going to spend a ton of time on this because we have done this quite a bit today. But in general, we have kind of created what is the criteria for biological hallmarks of aging that I think can start to address some of the limitations that something like a diagnosis or mortality rates cannot actually do. So it has to really predict a person's physiological, cognitive, and physical function in age in a related way. And it must be testable and not harmful to test subjects. So it has to be minimally invasive and something that we can collect in a least expensive way possible. But I think what's also important to this piece, or what I'm adding here, is this equity lens. And thinking about, well, should a biomarker of aging also be equally predictive in all subgroups? And how do we think about the between group differences in biomarkers of aging? Should they be exactly what we see in population health disparities more broadly, or are we picking up something totally different in the biomarker of aging space? So the things that I think are most valuable about biomarkers of aging in diverse populations is that, one, they get at more of the heterogeneity in health outcomes among especially Black populations. They typically have more variation than yes/no measures of health. They allow us to move away from this cumulative disadvantage narrative in some spaces, especially when we don't coagulate them all into one measure. They offer insights that often individuals can't report or don't report about their own health. And so they potentially can bypass some of the medical health norms that are more popular in white spaces, where people have a lot more access to care, are often much more engaged in care and feel more welcomed in that setting. And potentially that biases the data for Black and Brown populations who are not engaging with the health system in the same way. And they're also, of course, the earliest markers of disease and disability that we have now, besides social factors. So they provide points of intervention that we can potentially think about. So typically what we have done, now that biomarkers have been implemented in these large national studies, is that we create these risk scores. And most of that is done by creating dichotomous indicators of high-risk biological health and then combining them into an indices that creates one measure of biological risk. And so this paper up here, this is by Morgan Levine and Eileen Crimmins. This was one of the first papers coming out about race differences in biological age. You can see that it kind of mirrors what we think of like in terms of mortality. And as you get down to the higher age groups in the 70s and 80s, the difference in biological age decreases, just like some of the mortality crossover stuff that's happening with Black-white populations. And so I think it can also give us some insight as to what is happening with our oldest old and whether or not we really have what is the mortality crossover or selective mortality, where you get a lot of Black older adults who end up in this sample looking really biologically different than the Black people who have already been pulled out of the population at earlier ages. But I think where this gets a little hairy is where when we start to use biomarkers to inform decision-making and pulling them into algorithms or race norming that really gets things a little bit hairy. So for example, George Floyd's previous biological health and conditions were blamed originally for his death in terms of his autopsy report, that they've now reversed and blamed on the social factors, which was the police kneeling on his neck for 9 minutes. And so that idea of trying to blame the biology of Black people in order to create, ideas about what they deserve, what they have rights to, how much health care they get. Another example is the NFL. So they were doing race norming and skewing the data around cognitive impairment for Black NFL athletes relative to white NFL athletes. So they weren't qualifying for the dementia protocol and insurance-related care that comes with a lot of the NFL treatment that's going on around cognitive performance. A lot of interesting work has happened with the spirometer and the history of the spirometer in lung function and race norming in lung function as well. I'm sure you guys have heard of the pulse oximeter and the skin color issues related here. So as you can see, the applicability of some of these biomarkers is really where some of the discriminatory actions, I think, are getting picked up. And we're embedding a lot of this discrimination into the measurement, the biological measurement of what we're producing. So this brings up, there's a ton of ethical related issues when we think about biomarkers and social genomics in general. There's issues around consent, privacy, who's benefiting, and how we can share benefits within among the populations that we're researching. Ideas about data ownership and autonomy, consumer-based genetic testing and how that distorts the monetary relationship between the data and whatever it gets used for or sold to. But I think in terms of what I'm going to focus on today is really thinking about the applicability of this work to diverse and minoritized populations, and that being one of the main ethical concerns and how we should be thinking about biomarkers of aging. So you heard already about telomere length from Greg. Thank you for setting me up so I don't have to do that. I much appreciate it. But in general, we've been on the hunt for this hallmark biomarker of aging that takes in all facets of the aging process. And I think that's originally what we thought telomere length would be, this superhuman biomarker that wasn't just like blood pressure measuring one aspect of the aging process. It was really supposed to be taking in all, systematic changes in the body with over age, and also with social factors like stress processes, and also varied by socioeconomic status, race, ethnicity. So this was really positioned as one of the biomarkers that had the most potential within the last 20 years. But as we got into some of the data around this, I published in, I think, 2017. This was HR Health and Retirement Study data. And this is looking at predicted telomere length. So this is salivary telomere length coming from saliva. And these are gender stratified models with an interaction between race, ethnicity, and age. And you see for women on the left-hand side that Black women have the longest telomere length. They actually have the longest telomere length of both men and women, followed by Black men. And then Latinx women were right behind. They're in the green bar. And then white women have the shortest telomere length, and white men as well. The men are actually much more volatile than the women. I think a lot of that is sample size issue, especially for our Latinx men over here in this area. But with women, you see much more narrowing of disparities with age. Men, you don't see as much of that, but there's still a relationship here with age. But this is definitely not what we anticipated finding. And once we started looking across the literature, you know, we found that there was also a ton of mixed findings around race ethnic differences in telomere length. So this is NHLBI study showing that in general, there are longer telomere length in Black Americans compared to white Americans. There were a couple of studies that found shorter telomere length in Black Americans. The Diaz-Roof study is using MESA, and obviously MESA has their own data limitations in terms of cardiovascular disease. And then Arlene Geronimus found Black Americans also had shorter telomere length relative to whites, but that was in a Detroit area study, so a much smaller population base in terms of sample size. And then when you look at Latinx groups, there's quite a bit less data in terms of thinking about the telomere length among Latinx populations. But most studies found that there was no difference between Latinx groups compared to whites. And a couple, the Anna's Diaz-Rue paper from MESA found shorter telomere length in Latinx groups. So then we wanted to know, like, well, what does this telomere length, or Black people in general having longer telomere length, and then our Latinx group also having the same, how does that relate to health? So we looked at the biomarkers, all the other like 15 biomarkers in the health and retirement study, and physical functioning measures to try to get some idea like how this telomere length measure is relating to other measures of health. And if you look at our white group, you can see how much smaller the confidence intervals are, but nothing is lining up in terms of, so high risk health is in red. Ideally, what you would see is that all of these red dots would be associated with shorter telomere length, and these blue dots would be kind of moved over here with longer telomere length, because you'd have healthier folks along this in the low risk group. And that actually really doesn't happen. It happens a bit among our Latinx group, but you see how wide the confidence intervals are. You can also see how much more Black older adults are over into the 1.4 area, whereas white people are over there in the 1.2 area. So this isn't really lining up with what we think about health and aging in terms of telomere length, and is not lining up with any of the other established biomarkers of aging that we've already kind of set up. So then I wanted to know, you know, does telomere length explain any of these race and ethnic differences across these 15 indicators of health? It turns out it definitely does not, but I think it is important to at least look at some of the social disparities in the other biomarkers, since these are more established markers and often tell us what we think we're trying to find out about health and aging. So this is from also the 2008 Health and Retirement Study, and if you look at HDL and total cholesterol, there were no race and ethnic differences, but I think that is a lot. Those were dried blood spots, and so the dried blood spots had some issue in terms of pulling out the actual cholesterol levels that we think may be responsible for the lack of race and ethnic differences here. But for the rest of the markers, you see what we normally see in terms of disparities. So for HbA1c, you see Black and Latinx older adults both have higher HbA1c. CRP, Black older adults had much higher CRP. Cystatin C, Black older adults also had higher cystatin C or were at higher risk. Systolic blood pressure and diastolic pressure has similar trends. You see heart rate and then pulse pressure. And then when you look at some of the other physical functioning markers, BMI has a similar trend. Lung function has the opposite trend. And this actually gets into some of the history of the spirometer. This lung function was not measured by a spirometer. I think it was peak flow, exactly. But you'll see that the risk for lung function is much lower among Black and Latinx groups, which prompted the race norming in the first place in terms of qualification for services and beyond. So it's really interesting to start to pull some of these disparities out because they're not always disparities in the direction that you think. Walking speed, grip strength. Grip strength actually had the opposite look as well, except for among Latinx older adults, they have much higher risk for grip strength. And then tandem, balance, and cognition look similar in terms of disparities. I also ran a mediation model. So KHB, the decomposition method, is a mediation model for binary outcomes to see if telomere length, the variation in telomere length accounted for any of the variability across the race and ethnic variability across any of the biomarker and physical performance measures. And all of these are the percent indirects or the percent mediated by telomere length, and none of these were significant. The highest we got up here was for HDL cholesterol and total cholesterol, but as you know, there was no racial differences in those measures in the first place, and they're not significant. You get a little bit more here with lung function, which is interesting. Many people think that, at least from the history of lung function, again, that these are like biological or genetic differences in lung function, when lung function can also be calculated as a function of height, weight, and body size and beyond. So a lot of that isn't factored into some of this stuff. Then when we looked at stratified models to see whether salivary telomere length predicted any of these indicators of health, it did so for whites, but not in a way that's necessarily intuitive. So for HDL and total cholesterol, and then for lung function, the telomere length did predict high-risk functioning, but it did not among Black and Latinx older adults at all. So what does predict functioning, or what does predict mortality among Black Americans? Definitely not telomere length. You see it down here. It's doing nothing for us in terms of mortality. But it looks like a lot of the more traditional biomarkers are actually better predictors of mortality for Black Americans. So you can see pulse pressure, HbA1c, cystatin C, balance, and lung function are big predictors, and probably cheaper predictors of mortality that we can measure among these populations. So then when I looked across the literature to see what other studies we're finding in terms of telomere length and health, I mean, there are just as many studies who find a relationship between telomere length and health as there are with no relationship. And so the literature is incredibly messy and hard to understand because you find, one study that looked at 3 or 4 indicators, they'd find like one of them was a hit, 3 of them weren't, and then the next study would be that one was a hit, and it just is a mess to try to make sense of where in the health and mortality process telomere length might be lining up. What's interesting, though, is that same data was able to find quite a bit of relationship between telomere length and social factors. So there are some studies that use this exact same data and found relationships between social relationships, religious involvement, discrimination, depressive symptoms, lifespan adversity, and marital disruption. So it may be that these are just really noisy measures and that you have millions of variables in the HRS. Perhaps you can pick up on something and that's what's happening here. Or it might be that telomere length is really picking up on some social stress processes, but we can't quite figure out how that connects to health and well-being in any way. So how do we explain some of these mixed findings in telomere length? Jess did a really wonderful job of talking about the millions of spots that there are noise within telomere length data. A lot of that is due to methodological issues. Right now, a lot of what we know about telomere length is in cross-sectional data, and whether or not the continued investment in telomere length will continue at a population level, I think is still up for grabs. It may be more important to think about intra-individual change rather than absolute telomere length. That might be more of the key, and so longitudinal data is going to be really important in thinking about some of those things. Cell type, as we discussed, was an issue. All of this data is from saliva. Assay, storage time. Jess actually did this study where they looked at storage time of telomere length over 12 months. And you can see this change between 6 and 12 months. That really changes the telomere length. That might have some impact on what we're looking at as well. And when you're thinking about like the non-random distribution of disparities and how going out and collecting data in certain places over others might change, storage time, time to assay, all the things that may impact certain types of communities rather than others could be embedded in this data as well. Another really important piece that starts to move us into the social genomics sphere is this concept of ancestry, and moves us out of this, traditional biomarker conversation. Telomere length is a highly heritable trait. Estimates range from 0.36 to 0.82. And we now know and have found that Black Americans and anybody with African ancestry has longer and is born with longer telomere length. So quite a bit of work has been done across sub-Saharan Africa that has basically shown that Africans have longer telomere length than both Europeans and African Americans. So there is also something happening here in terms of our thinking about racial disparities that's happening around heritability. This graph over here is showing mean average telomere length, a bunch of tissue types for European ancestry and African ancestry. African ancestry is here in red, and in general, you can see telomere length is longer, regardless of tissue type for African ancestry populations. So one of the things that I think is really important to discuss is this concept of heritability and what this really means when we say something is a highly heritable trait, especially as it relates to race and ethnic health disparities. So heritability is really addressing like a population level correlation between the phenotypic variation and the genotypic variation in a population. And if heritability is high, that means it's least influenced by population or by environmental traits. If heritability is low, that means it's highly influenced by environmental traits. But I think the important piece here for disparities-related conversations is that heritability can change without any genetic change occurring in a population. So when an environment starts contributing more to variation, that heritability level is going to change regardless of whether or not actual genetic heritability or passing from one generation to the next changes. And I think that's a really important point to consider when we're thinking about health disparities, because oftentimes we think of genetic factors or heritability factors as kind of these hard truths, these real truths that stay constant over time. But they're very much influenced by the environment, and that can be different for different populations, even within one country, that amount the environment is playing in that role. So some of the consistent telomere findings that are emerging, we know that telomere length is longer in men than women, and we also know that it's longer in African ancestry populations than European ancestry populations. Telomere length in general is correlated with age in most tissues. It's influenced by genetics, heritable, non-genetic maternal and paternal factors, and environmental factors. There are a couple of interesting disorders associated with extremely short telomeres as well that are clinically relevant. So really short telomeres in a clinical setting can mean something different in that, there's some medical genetic disorders like bone marrow failure and lung disease that are important for telomere length. We are also picking up some noise in terms of lung function among the white population with telomere length, so there might be something happening there. And then we also know that there are longer telomere length for cancers, for certain types of cancers. But there's a whole host of quandaries still in the telomere length research. One is that I think one issue that really affected the initial emergence of telomere length into the biomarker space is this media hype and premature commercialization of telomerase. I think geroscience is one of the spaces that's most prime, as Jessica said, to make it to the consumer space as a result of the biotech industry. And sometimes that can really interfere with the quality of what the messaging is around some of the science and how that's really related to health. How does race ethnicity interplay with some of these ancestry differences that we're seeing in terms of African ancestry and admixed populations? We've had problems translating, as Greg so brilliantly pointed out, translating from animal models to humans and from small homogeneous samples to really large diverse populations. It's a really noisy measure, and causality is very difficult to establish. We talked about the literature being very messy and publication bias towards significant findings. I was lucky to have mentors who pushed me to publish null findings, essentially, around telomere length, thinking that that was just as important as the findings, the positive findings. And as a result, it's really influenced how I view the publication process and beyond, but it's important to push those findings forward as well, especially with new ideas like biomarkers. So we're publishing both null and positive findings. And then where's the utility for this? Is it in basic science? Is it in clinical settings? Or is it really going to be in or population health? I think all of that is still to be determined. I'm not going to spend too much time on this. Coulter always spends time on this, but I think telomere length is somewhere within this overhyped expectation space. We're trying to get down to figure out whether or not it's going to have any value for us, and whether or not it will have any value for race and ethnic health disparities more generally. So I think it makes a lot of sense to also pull this discussion into a space of what's happening in the genetic sciences more and genomic sciences more broadly, which is that we are primarily training all of our genetic data in European samples. This chart shows that, majority of our sampling is happening in North America and Europe. Very little of it is happening in Africa, which I think is incredibly relevant to this discussion and thinking about the dynamics of the African genome as it relates to African Americans is a really important conversation to be had as well. But the fact that we don't know that much about the African genome is going to definitely affect African Americans in the United States as a result. And then you get into this algorithmic issue where you start doing all the data reduction strategies and applying it, and the same conversation around the biomarkers of aging that we already had with the NFL and beyond, and spirometers. The same thing is happening with a lot of the algorithmic bias that's being built into something like a polygenic risk score or an epigenetic clock, where now you're embedding some of this bias in the data into an algorithm that can further produce racial inequality. Some people think it's maybe an overstated claim to think that inequalities in health could be exacerbated by this problem. But when you start to think about something like pharmacogenomics, that's really going to take off as a result of a lot of this work. That's where you really start to see the application of this not being applied equitably. And this equity, I think, is really the larger issue here that we need to confront. So when we think about this equity issue and empirical ethics, I think there's a lot of things that go on into this conversation. One is that we need to continually reassess the value and impact of genomics and molecular biomarkers in Black, Brown, and Indigenous populations. A lot of the ethical and empirical work done in social genomics is coming out of the indigenous communities, at least in America. There is also conversation happening in terms of colonialism in Africa and how the genome is often assayed and data is collected and then moved back to the Western world. And so there is also some of that conversation happening there, but a lot of it's really happening in indigenous populations. There's also something called genetic data capitalism that has been this kind of idea that certain people benefit off the genetic data space, and who are those people, and centering that conversation around who should be benefiting from genetic and biomarker science is really, I think, an important question to keep asking. And should we continue to upscale this collection, knowing the dangers it plays in terms of equity? So that brings me to this question around, what would it look like if Black people owned their own genetic data, or if they were involved in the concept making of owning their own biomarker and genetic data? What would that really look like? There's a really, I don't know if you guys are familiar with the ELSI. It's the Ethical, Legal, and Social Issues in Genomics. Really wonderful group ran by the NIH, one of the genomics, I think it's the Genomics Institute for the NIH. And, they're starting to think about these questions, like what would it look if Black people were starting to be involved in this concept making around social genomics and around biomarker data? And I think first and foremost is that it would be integrated into social stress frameworks. It wouldn't be above social stress frameworks. It wouldn't take precedence. It wouldn't be center stage. We would always acknowledge, that health disparities are really the result of social and physical environments. And acknowledging also the history of exploitation around some of this data is part of this acknowledgement process, I think. And thinking beyond the biological concepts and thinking about social histories. We also, I think, need to be resolving the social inequity first. Social genomics would have a much greater impact in a more equitable aging landscape. And if you don't address some of these social inequalities first, genomics is not going to have the same effect for all groups, and that's going to impact what we end up coming out with on the back end. The history of ancestry. This is not just a biological concept for many people. It also is a cultural concept, and it has a history that stretches beyond, the beginning of the biological testing of ancestry. And so acknowledging that identity and cultural ancestry can also be a part of this conversation, and maybe should be informing how we think about genetic ancestry as well. We talked a little bit about the Black American ancestry and its connection to the African genome, making that a real explicit intention for Americans to think about the African genome as it relates to the people who come from Africa and are in this country. I think that would be a really important step. And then thinking about, of course, data ownership, privacy, benefits, justice, transparency, accountability, sharing a lot of the benefits that come from a lot of this, I think is really important. And at the end of the day, thinking about the equitable solutions for the benefit of Black people is the larger aim of this work. This is a really great webinar if any of y'all are interested in the decolonity of genetic ancestry. And finally, what are some of the things we can do to improve the measurement and application of biomarkers of aging in unequal societies? I think having this conversation in general outside of Black communities in and of itself is potentially an issue, right? We should be involving them. They should be a part of the process, engaging them beyond. But if we increase studies done with and not in, not for, it's with underrepresented groups, with them at the table, is part of that conversation. Integrating studies focused on social determinants and having these broader social models of disease, like Jessica so wonderfully pointed out, that are we really even explaining that much variation in terms of disparities? And so then you really have to think about how these types of markers are really integrated with social determinants of health. Considering the variability, the genetic variability within ancestry groups, why are we using certain population descriptors, and defining why we're using them and when we're using them. Building infrastructure and transparency, not just building knowledge and data literacy, but building the infrastructure and transparency in communities that are interested in participating in genetic research. Having a full appreciation for the limitations of biomarker and genetic data. This is something Greg and Jessica did flawlessly, was making sure that we're appreciating how limited this data can be and acknowledging that up front, and then adjusting our what we're saying about the applicability of this data as a result. Keeping human feedback in the decision-making loop. I think that's really important too, rather than just embedding the algorithmic biases continually. It's important that we have this reassessment and prioritization of what's important in terms of these data reduction strategies. Enhancing trustworthiness of biosocial and genetics research in these communities. That's thinking about benefit sharing, autonomy, productive data use agreements, acknowledging the histories and power dynamics in data collection. And also, and a really important point that will be my last, is that less than 4% of the tenure track faculty in research-intensive biomedical departments are African American, Latinx, or Native American. The training for a lot of this work, I think, is a bigger conversation that needs to be had about how we're training folks to do a lot of the social science work, and how can we get communities of color to be engaged at the epistemological level, so then that can all trickle down, and the types of questions we're asking will be a reflection of the population, of the science population we're recruiting. Acknowledgements. I want to say thank you to the diverse group of older adults who continue to participate in the health and retirement studies so that we can all have these wonderful careers. Folks at USC, some of those are outdated in terms of location. Twitter handle is down there if any of you guys want to connect on Twitter. And then I added a couple resources for anybody interested. There's some really great pieces now about the African genome and the role it plays in terms of thinking about the global genetic issues we're having. And then some resources around data capitalism and how genetic data may really be wrapped up in the data capitalism space. LC Hub, great resource. Definitely check out the LC lectures. They're wonderful. So thank you all so much for your time. I appreciate it.
Thank you so much, Lauren. I'll ask all three of our presenters to sit up in the front, if that's okay. I know we're just about at lunchtime, but if we can all stick around for a few minutes for some discussion and conversation, I think that would be great. But if people need to leave, we definitely understand. So questions, comments, thoughts? Also to our friends on Zoom. Yeah.
Okay. Thank you all so much for the really great talks. I had a question for Lauren. You brought up several examples of kind of like racial difference in the methodological processes that occur in like the collection of these samples. Like, have you seen any evidence for that in the biomarker that you were talking about today, telomere length, and then also with regard to the telomere length differences across racial groups, you mentioned that as well. Does that, how is that incorporated into an analysis of telomere length in these diverse samples?
Thank you for those questions. So if I'm hearing you right, you're asking about how data collection processes may have impacted like racial differences in telomere length. I think that's maybe something we don't know quite a lot about. I'll just say I don't do any data collection. This is all secondary data done by the HRS. So a lot of what I learn is really from Jessica Coulter, Eileen Crimmins, people who are really involved in trying to create the data collection landscapes that we all use in order to be able to like create our careers and move forward. So I think, my role as a social scientist and a junior social scientist is coming in, getting the data and wondering what I can do with it in terms of, my research agenda. And so when I started the project around telomere length, I really wasn't thinking about all the roadblocks. I was just thinking, oh, hey, let's connect this to social stress. And when that didn't happen, that's when all the backwards movement changed. Like my movement in terms of how I was thinking about this totally changed. And then it was like, well, then what are the data collection processes that could be impacting these groups? And I don't know, Jess, do you have any hunch about what some of if the data collection issues could impact racial differences? I think one thing I'll say is that, we see all these issues with telomere length. One thing that does not create some aspect of issue is that with racial and ethnic differences, we're looking at relative differences. So the idea of like cell and tissue type technically should not be different in terms of comparing populations. And I don't know if you agree with that, but this idea of like relative telomere length in thinking about white, Black, Latinx, we're removing some of the variability and noise that would be embedded in the entire population anyway. So we're just looking at like, what this would be like. So I think we can really assume that the methodological issues may not be the total source of a lot of what's happening in racial differences in telomere length. Do you have anything?
I think in this particular case, the data collection issues are probably not what's going on in terms of race ethnic differences. We have the same collection protocol across everybody, and I don't think there's a these are bulk samples. So as Greg showed, we've got all different cell types in saliva and all different cell types in blood. So if there's anything that changes the composition of those cell types in an individual, then we're kind of averaging over different types of cells. And those different types of cells could have different telomere lengths, or different DNA methylation patterns. And I think that's more likely to be what's going on because we're not isolating or really adjusting by cell type for most of these things. So, you know, if you've got a lot of inflammatory processes going on, you might have a different composition of cells, or like Greg said, those cells could be working differently. And so I think that's more likely what's going on with these race and ethnic differences than anything like wonky in the way that we're collecting the samples. Oh.
That's great. That's super helpful.
Thank you. Paul, did you have a question?
Yeah, I guess it was a comment. Thanks. So I think a unfortunate kind of mathematical nuance in a lot of this stuff has to do with what Lauren referred to as, you know, are these things equally predictive? And then it came up in Jessica's talk too with looking at differences in correlations. And all these things are measures of association. You could talk about correlation, odds ratios, mean differences, whatever. And whenever you're talking about those things, the scale of that thing really matters mathematically. And it depends on what you're going to apply the biomarker to in terms of which scale is important to you. And, or another way to say it is, you know, some people have shown that, you know, detecting interaction effects matters, the scale of estimation matters for that. And so it isn't so much a question, it's just a comment that we're looking across these groups, we're looking at differences of association, and the metric of association, unfortunately, sometimes can really change the answer that we might get. And so that's an added complication. That's all I wanted to point out. And I saw that theme across all three talks.
Thank you. Other questions, comments? Yeah, Bridget.
Thank you guys so much for such a wonderful, informative set of talks. These just came together beautifully. Oh, I think that microphone might be interfering. And so this is to build upon what kind of what this last comment was. Another thing that I started thinking about and listening to all of these talks is time and the role of time with these with markers, right? Because a lot of this was, you all said, was cross-sectional. And so how are you thinking about, for people who are collecting biological markers, y'all know me, so y'all know we are doing primary data collection, but in thinking about time, but like how would researchers who are not collecting their own data, but using these pre-existing data sets can think about time and the dynamics within which we're measuring these markers at a particular moment where we don't know all of the information about a person. We don't know all of the context that's going on around them, what's going on in their body across multiple systems. It's complicated, right? And so just how do y'all kind of think about addressing the dynamics of the body and the dynamics that intersect with time when we're thinking about these biological markers?
It depends.
You can't get your mic.
I mean, I think it depends on the biomarker. I mean, some of these things change, I mean, like cortisol, it changes throughout the day. Other things like DNA methylation, I don't expect to change, like even across, substantially, even across years, unless you're a fetus. So, you know, I think again, I mean, this actually gets to the scale issue too. I mean, the extent to which any of these markers is going to predict a longitudinal outcome also depends on what you're predicting and how changeable or dynamic that process is. And there isn't a, you know, set answer for any one of them. You know, but you all know from getting your own blood pressure measured, like just how volatile that can be in the span of 5 minutes. You know, so I think it's complicated, and you know, it's complicated both in the measurement of it and, as Lauren talked about, in the translation of it to clinical practice. So I don't have an answer for you.
I think I'll comment too. I think that I've largely ignored this issue by virtue of thinking like, you know, if we catch Black older adults after they've been lived through, you know, when segregation and hospitals and education being segregated, to the civil rights movement, to, you know, Barack getting president and the Black Lives Matter movement, by the time we capture them in their 60s, 70s, 80s, ideally we're eliminating some of that issue with time. but that's actually not true. Like that was coming into it, I was thinking like that will be the best time to think about and measure these biomarkers of aging, thinking that the closer we are to death, the closer it would predict death and beyond. And really that doesn't work out in the biomarker space. And I think that concept of time is totally warped in the sense that we normally think. And something like chronological age is not helpful in thinking always about the biological age, because time does not follow that same format. And appreciating that, I think, is the bigger issue of what we're here to do. I mean, that's exactly the context in which we're all trying to work, is to better capture what isn't just time, what is more actually this biological process that may or may not reflect time. Could be totally different, or it could be very aligned, one of the two. Great question. Mm-hmm.
Okay.
I'll just add briefly is to go back to my last example with the multiple layers problem and cortisol and immune cells, is that the subtlety I didn't convey in that talk is that when we followed those caregivers for glioblastoma patients over time, all those systems moved at different paces. So it turns out that shortly after the initial surgery to remove the tumor, cortisol was up in the caregivers. It just normalized very quickly. And all of those markers changed at different paces and on different trajectories. They're all interconnected talking systems. They didn't change in ways that I completely understand, but the idea that biomarkers are static, or even that the relationships between biomarkers at different levels are static, is also probably a fragile idea. And it speaks to, I guess Lauren isn't here at the moment, but oh, there you went. But you asked me and all of us to talk about the interplay between systems, which I've kind of failed to do. But I think that's another important point about coming out here, is that if you really take a broader look, like we all tend to silo in on one biomarker that we want to get famous for, we think is important. But when you take a broader look at multiple biomarkers, they often don't converge. Even when you're looking at like protein levels of inflammatory markers and messenger RNA and epigenetic, presumably the same thing, they don't converge. And so that's where time gets even trickier. So.
Yes, I have one question that's a little bit open, but actually, Lauren, you pulled from a couple of studies I'm going to kind of talk about in a second, especially the living study. But one thing I keep kind of coming back to and thinking about is a lot of the biosocial research, right, is really emphasis kind of on the individual systems and those changes within those people. And I'm thinking also within the larger context of what's going on in the environment. And I'm going to be a little more specific here. So with the Levine study, you showed, well, that study, you didn't show this, but that study kind of showed that biological aging between Blacks and whites in the United States was kind of explaining mortality differences. And I just had a study that was published 2 months ago that actually shows that Latinx older adults have some of the highest biological aging. But then we have I hate this term, but the Hispanic paradox. So I often think about how these might be differentially predictive of outcomes based on partly the context in which people are embedded. And the best example I can think of this is I was writing this in December of 2020 when those COVID death, right, the loss in life came out, and Hispanics just dropped. By 3 years, we know it's highly, right, it's linked to immune and aging. So I thought, so for me, it was kind of no surprise. Yes, it is in terms of, right, the exposures, things like that, but we do see that difference. So I just figured kind of drawing it back is how do we think about even the larger context in which individuals are living when it comes to biological risk and some of these measures.
Yes, I actually think bringing up the pandemic is a really good point, because I think it's something we all experienced, and but we experienced it differently, right? And so when I think about collecting biomarkers now in 2022, part of me is really sad because, my time 1 and time 2 has the pandemic in the middle. If I want to understand what normal aging looks like, well, I can't now. But in reality, that's happening all the time. It's just that this is a context we've all seen, but these contextual factors are there constantly, all the time, differentially affecting different groups of people in different ways. So I don't even know that there's like normative aging anymore without the social, behavioral, the environmental. So I don't even know what's normal, but I think it's a really good point that the context matters, and the context is largely what's contributing to the heterogeneity in our ability to understand and then use these biomarkers and predictive models or to understand biology. I think you didn't, getting back to the question I think that was asked before, or I mean, maybe what Greg talked about, I think also, you know, we just have to have an appreciation that these are dynamic systems, you know, that our bodies aren't designed to go haywire and stay that way. They're constantly trying to fix themselves. So I think, you know, thinking about, you know, again, we constantly frame these biomarkers as things that are bad, or in reality, it's a really complex and dynamic process, and it's a really good thing that our bodies are really good at fixing these exposures. So I think, you know, we also have to kind of reframe some of them a little bit too, and thinking about them as sort of biomarkers of health, of our bodies doing what they're supposed to do. It wasn't your question, Mateo, but.
Mateo, I loved your question. I think what your question highlights is really that, like, the cultural lens with maybe how we think about some of these biological and genetic markers may need to be even more serious than we've given credence to. And, you know, race and ethnicity, I think, can be a contextual or social-political contextual factor where we're trying to do that. And the social sciences, I think, in the health disparities space, has really tried to move away from race as the way of, you know, marking these disparities, and say, no, it's not race, it's actually racism, right? And perhaps the biological health movement will move in that direction in places like the HRS, where there is this dynamic linkage between you know, social environments and racism and, you know, some kind of biological outcome. And I think that may need to happen for the correction around the discussion of what we're having in the social and biological sciences, so that the blame isn't placed on the race and ethnic group. The blame is more attached to the social environment that we're discussing. And can we be more accurate in that discussion, more accurate in the predictors? Race and ethnicity, I think, is a really crappy social category that's really getting at environmental differences in health. It's getting at differences in stress experiences. It's getting at a lot of different important factors that we've lumped into one group and tried to use it as like a shortchanging way to get at social disparities. But we can probably do a better job at getting to the point of which, like, well, what is it about race and ethnicity? And how can we better tie that discussion so that it doesn't get lost in translation down the line in the algorithmic space or the application of some of this work? I think that's especially relevant. Like, I'm teaching an undergrad stats class, and they're learning race and ethnic health disparities. And it's a nightmare to try to get them to move away from some of the language that they have learned about race and health and public health for the first 4 years. They're seniors. So that first, you know, 3 years where they're really getting embedded about race, ethnic health disparities, and this focus on Black, white, Latinx as really the conversation, when perhaps the conversation really has to be moved to more around discrimination, environmental racism, structural racism, and beyond. Thanks.
Thank you. Any other questions? Rita, are there any questions in the chat? Oh, Connor.
Thank you all. Jessica, this question I think is primarily for you. I was wondering if you could talk about how these epigenetic clocks that you've discussed perform differently or perhaps similarly across different developmental periods.
I don't know yet. I only study older people. You know, the clock, I think it's important to remember that the clocks are designed to do the thing that they're trained to do. So my guess is that if you looked at younger groups, you would see some of the same differences, you know, so you would see a range in sort of the age acceleration. But it probably looks different depending on what that clock is trained to do. So what I mean by that is, if the clocks are working the way we would hope they would, so a clock of mortality, you would see maybe more heterogeneity and a larger range among older people, right? If they've accumulated a lot of stuff in their life that, makes them more or less at risk for mortality. Whereas in kids, that's going to be a lot tighter. It doesn't always turn out that way. And again, but it's largely to do with the clock itself and what that clock was trained on. But I think I think this is going to help us understand a lot about epigenetic mechanisms as we take not just the clocks, but there's other DNA methylation surrogates that we use to predict lots of other things, and EWAS, which are predicting sort of the biological side of, predicting disease or behaviors or whatever it is that you like, and actually applying these in different age groups and seeing what their relationships are, I think will help us understand, is epigenetics or is DNA methylation something that is set in utero and then we're just on a course for the rest of our life, or how much can it be impacted by factors that we're experiencing in our life? I think taking all of these things and looking across the life course is exactly what we need to do.
Oh, do we have time for one more question?
I was just curious if you could talk about some of the most interesting or important sex differences that you've seen in like measures of stress or biomarkers of stress, or even like responses to stress between sex.
We don't see much. I've got to say, I know there are a lot of folks who say that the effects are massive and that they see them everywhere. We just cannot in any of our studies reliably. Maybe we're underpowered. Maybe we're not looking in the right places. But I got to say, we mostly turn up nothing. And this is in studies of disparities in asthma, disparities in perinatal outcomes, disparities in placental gene expression. Like, we just don't see sex differences in the strength of the association between various measures of adversity and health outcomes. I've been stunned at how strongly people believe. And my read is that there's the sex differences are quite strong in mice and in non-human primates. And I think it's a truism that mice lie and monkeys exaggerate, as they like to say in biomedical research. I'm less persuaded that in human studies, the differences are as robust, at least in terms of kind of the peripheral immune stuff that we do. I don't want to go into the brain or other places, but I'm not knocked out by it.
I wonder too if we're measuring sex in these studies. I'm not sure we're measuring sex in a lot of the larger studies that we're looking at. I think, it depends on what you mean by sex. Is it like an anatomical feature? Is it hormone levels? I think we're starting to think about sex and gender as a spectrum, and how we've traditionally modeled it in large population health studies is something that maybe needs to be reconsidered, maybe entirely and altogether. And thinking about sex differences in terms of social stressors, that question in general may need to be reframed in thinking like, how something of like a biological category, if we mean gender differences, I think gender and social stress are more of the connection here that we're trying to draw around, like what is a woman's experience based on her experience in the world? And as a result, we think about, the social stress that comes from being a woman walking around in the world. Rather than thinking about like the biological features of sex. If we do think about biological features of sex, then we have to really be measuring what's underneath the biology of that anatomy or beyond. And I don't think we're doing that right now at this point. So.
Excellent. Thank you. Thank you for all your questions. Karen?
Yeah, I just wanted to wrap up by saying Deb and I had the privilege of witnessing as Lauren was planning this, and each of you said yes, and we just got more and more excited. And I can't believe that you surpassed my level of excitement. Thank you. You were just amazing. I think I'm speaking for everyone here. And I also wanted to say that Lauren gets so much credit for the extraordinary job of organizing this. So thank you, everyone.
Thank you. Thank you all for coming. Thanks again to our panelists. I had this vision of how well these talks would go together, but really you've exceeded all of my expectations. They were beautifully linked and drew on different areas of inquiry and problematizing our thinking around biological aging. So I really appreciate all of you. Thank you so much for coming. And if you will please join us, we have lunch. Rita, is it just next door? And you are all welcome to join us. Thank you again.